All right, we should be live. I'm Tad with KIS Organics. I'm here with Bryant Mason, Soil Doctor Consulting. And we're hoping to do a short, 20, 30-minute live every week here on YouTube. And the topic this week that we came up with was microbes. Bryant, and how it relates to soil health, soil management, and those sorts of things. Bryant, do you want to give a little bit of what you've been thinking about in regards to microbes?
Sure. Yeah. We have these conversations all the time, Tad, kind of offline. How I think about microbes is really from the standpoint of, a crop advisor, where I'm trying to figure out when different management actions make sense. So, does it make sense to apply microbes? How do different nutrient inputs sort of like affect microbial communities? And so um, when the soil food web was getting really popular years and years ago, I went deep into the science of trying to understand soil microbiology. And I mentioned this book on my Instagram earlier. This to me is like the Bible, at least my Bible, when it comes to soil biology. And I read the thing cover to cover and just took a ton of notes on sort of the high level concepts. And the thing that I think about most is what this book sort of says is that the soil habitat is what ultimately determines microbial populations. And that's kind of interesting to me because it's a little bit of a different framing from what microbes should we be adding. Because to me, what actually matters is what we should be doing to the microbial habitat to then influence which microbes are going to exist and in what quantities. So,
Well,
yeah, that's where
let
I'm
me
at.
ask you a question then about that, Bryant, because the soil food camp would say that the plant controls the microbial communities in the rhizosphere, and it's able to dictate, what communities are going to be more successful based on the exudates it's putting out, based off of, gas exchange. And yeah, I mean, primarily those two things, I would think, in relation to moisture is what's controlling the microbes. So they would say it's not the soil, but the plant. Like, how would you look at it?
Okay, so I would say that there are three primary chemical factors that influence microbial populations and three primary environmental factors. And the plant species is one of those environmental factors. So plant species affect microbial communities by releasing different suites of compounds into the rhizosphere and during tissue decomposition. So this is from simple organic acids to really complex secondary metabolites. And plants also interact directly with microbial symbionts and through different compounds, but that's one method. So to say that that is the primary variable, I think is a bit inaccurate and oversimplified. The research, at least according to the research, in this textbook, is saying that soil moisture and soil temperature play a far greater role in influencing microbial communities. So anyway, that to me that's where I'm at. And then when you dive into the chemistry of it, soil pH, soil redox, and soil aeration are going to drive microbes far more than just, plant species interactions alone.
Okay, well, do you want to touch a little bit on each of those?
Yeah, for
And what
sure.
their role is in sort of microbial health?
Yeah, definitely. So soil pH affects a number of factors, around microbial activity, such as like the solubility and ionization of different ions. And it really changes the soil solution constituents, which in turn affects soil enzyme activity and ultimately microbial populations. So, yeah, I mean, microbe populations between us with a soil with a pH of 5 versus 7 are going to be wildly different. They're all adapted to their different ecological niches, and pH is a major influence there. This book also made an interesting point. This is just an aside, that there's sort of this general belief that, and this is what I always thought, it goes back to this old scientist who just suggested that everything is everywhere, but the environment selects, meaning microbes are sort of everywhere. And so I always thought that the same species of microbes would be sort of in all soils, but the data suggests that endemic species are more the rule than the exception. Endemic meaning that these species just live in a particular ecosystem. So an endemic species to like the Columbia River Basin it only exists in that river basin. It doesn't exist anywhere else on Earth. So there's a lot of that happening in soils where these conditions create these very unique microbes, or what people call indigenous microbes. And so I tend to agree that indigenous microbes are better to cultivate because that's just usually how it works in the environment, because all of these things influence microbes. So, okay, pH and then redox, soil redox, which is complicated. Essentially, the reduction and oxidation reactions in a really saturated soil, for example, you're going to get a much more reduced environment. And then soil aeration. So how much oxygen and gas exchange is happening through a soil. So those three things influence microbes wildly in terms of like the soil solution. So these microbes mostly exist in pore space in the soil solution and on the surface of minerals and different organic complexes. And so what happens in the soil solution matters a lot. Another interesting thing that people should understand is that the soil solution is not homogeneous. So even though we test things and we say, well, this is what the soil solution looks like, it's actually that one pore is going to have potentially soil solution that has maybe a slightly lower pH and higher salinity than another pore. So there's definitely diversity in the habitat. There's no doubt about it.
Yeah, that's a really good point. I heard a talk by a microbiologist talking about this in relation to soils, that there's all these little micro pockets where you have anaerobic microorganisms and aerobic microorganisms. And because, I mean, we describe it pH and, EC and all of these things as being sort of homogeneous in the mix, but really it's not like that at all. This is like a huge ecology with a lot of little variation going on all the time in there. And one interesting thing I want to point out is, years ago, I got to attend the Soil Scientists Society of America international conference in San Francisco. And there was a presentation on essentially looking at how microbes fluctuate in the soil based off of hydrology. So what moisture level there was in the soil. So they measured they looked at the fungal to bacteria ratio. They looked at what species were in the soil and what biomass levels they were all there in. And what they essentially found was that when it rained or when there was a drought condition, so just a typical fluctuation that we would see normally outside, you had a massive variation in fungal to bacteria ratio and in the microbes that were present. At least the concentrations of those microbes would vary so much, which when you think about it, makes a lot of sense. At least it did to me. And so that has sort of moved me away from using fungal to bacteria ratio as a metric for evaluating soil health, I guess, in general.
Yeah, no, that's a great point. And that's, like I said, soil moisture is one of those environmental conditions that matters a lot. This book said that generally aerobic microbial activity is maximized when, and this is topsoil, right? So when we're talking about soilless media, this is going to change, but it's considered optimal when the water potential ranges from negative 50 to 150 kilopascals. So people who use tensiometers and things, that's kind of an interesting note. Big range, but still it matters. When your soil dries down beyond that, microbes die or go dormant. And yeah, what were you gonna say?
I don't know. I lost my train of thought there.
Yeah, that's right.
I'm struggling a little bit today with this head cold, I'll be honest.
Yeah, so I was going to say, I listened to this great podcast, a Huberman podcast on colds and flus. You should check it out. Very, this is a tangent, but.
Okay. Yeah.
Yeah, you should, There's good tips in there about staying healthy.
I could definitely use that. But getting back to the microbes thing, I did want to say, so speaking of this whole idea of fungal to bacteria ratios, I think this all started, I want to give a little background here. So where I typically saw that is Jeff Lowenfeld's book shows this microbe succession or this plant succession chart, essentially saying that when plants first come onto bare earth, you have your lichens and lower lower organisms, and that soil is going to be highly bacterial. And then as you work your way through grasses, they find the soil tends to be more 50-50 fungal to bacteria according to this chart, and all the way down to an old growth forest where the soil tends to be highly fungal as it becomes more established. And so people will say, okay, I'm growing a cannabis plant or I'm growing a tomato and it falls here on the chart. So I want my soil to be approximately that fungal to bacteria ratio, with everyone assuming that bacteria is going to be flourishing. We need to raise fungal levels in soils. So that's where I think a lot of this idea started, of looking at this ratio and trying to raise fungal communities in soil.
Sure. Yeah. I wonder if people are sort of confusing the cause and the effect. The reason I say that is I want to see the evidence that a fungal dominant soil necessarily produces a better crop, for one thing. I do believe that, and again, what the science says is bacteria to fungal ratios do tend to be lower in acidic, low nutrient soils with a lot of recalcitrant litter and high carbon to nitrogen ratios, like a forest. And bacteria are increasingly, dominant in high nutrient soils, saline soils, alkaline soils, and so it makes sense that those conditions, again, the habitat is what's dictating a more fungal or more bacterially dominant soil. But to say that one is preferable for crop production, I'm not even totally sure about that. And the other thing is, I just think trying to change that ratio with inputs isn't the best solution. You have to create the environment that I just laid out in order to have a fungal soil, which would mean much lower nutrient levels, a lot of, decomposing carbon like wood material. And you do all these things, which could definitely produce a fungal dominant soil, but in doing so, are you really optimizing for the crop, or are you just trying to create a fungal dominant soil?
Yeah. So when I think of microbes in relation to soil health, the main thing that I'm looking for is nutrient cycling. So that's what I think. I don't worry so much about having this one particular bacteria or fungus, or even what that ratio may be. But I want to make sure that whatever's in the soil, in theory, is just cycling nutrients from my plant. And that's, to me, the biggest benefit of microbes and having good soil with good microbial health. The other thing in relation specifically to cannabis is this idea that, and this is getting a little bit more speculation on my part, but I truly believe that what separates living soil from chemical growers is that these microbes are what's releasing some of the unique terpene and cannabinoids there's things that we can't replicate just by giving a plant constituent elements that it needs to stay alive. But again, I don't have any proof of this, but that would speak to why in the market these days, living soil, weed is so much more popular and valued higher, I think.
Well, that's 100% the reason why I've sort of been drawn my entire life to organic production and living soil and just a very biologically rich environment. Is I do think that it's not just biology cycling nutrients, but the exudates and the byproducts of dead microbes and microbial just necromass and things is what creates amazing secondary metabolism in plants. So I do think that there is a potential advantage there. I just think that we should be really managing the habitat and trying to abide by principles that maximize biology in terms of, using carbon. So keeping carbon flowing into the system. I do think minimizing disturbance is a really good way to maximize microbial diversity, which can be difficult, but I think that is a good practice.
I think that could be our next live. I have a little bit of a theory around this, talking about ecological disturbance that Ben Higgins got me thinking about. So I'll save that for the next one. But I'm now at the point where I think some level of disturbance might actually be really beneficial towards microbial diversity. But what you're saying is, I totally agree with this idea that we're going to manage our soil with microbes in mind, regardless of what, we don't need to know what those microbes are. We're going to treat our soil, these are just good management practices. Like that, like for me, like biodynamics is not my thing. I don't know that burying a cow horn is going to make a big difference across a wide acreage space and all the other things that they do. But I think the practices that the soil management practices in general that biodynamics brings to the table are just really, really good for soil health. And so that's why I think it's so effective. So it's the same concept for me, I guess, when I think about microbes. Like, I'm going to do the same thing regardless whether I'm testing for these microbes or anything, like you said, the mulching, the moisture, the right pH, the right
roots in the ground.
All of those things.
I mean, the best thing to keep microbes alive and cycling is roots. So yeah. Now, yeah, go ahead.
Oh, I have a question for you. So all that being said, I think, and I want this to be interactive, and I'm seeing the comments popping up over here, and we can address them individually, but I think the big question here is like, okay, so how do we take this and apply it to our garden or our soil? So, you're going to go to, you're going to see on Instagram or on a hydro shop or whatever this, there's this particular Bacillus species that is known to do this in research. Should I buy that for my soil? What products on the microbial side may or may not be worth it?
Yeah, no, and I was hoping to bring this up because it's sort of, so this is what the research says: the habitat is all that matters, and that that introduced microbes have an extremely short half-life and will get through both competition and getting eaten, like direct
prey predation,
trophic interactions. That said, I still use them on my own operation. I mean, I bought microbes from you actually and inoculated my trees with mycorrhizal fungi. So I think that there are uses of inoculants. First off, I think seed treatments have been proven to be very effective. I think that mycorrhizal fungi applied to the root at time of planting makes a big difference. Or at least the research shows it can, especially when the plant experiences stress. And sometimes colonization won't happen naturally. So I think those, and I also like with my annual crops, I do a root dip with Taneo Spectrum plus Myco, which I sell. So I think there are times that inoculation makes a ton of sense. And I think what happens a lot in high intensity production, specifically cannabis, is if you slam like trichoderma, for example, consistently through a round, those species will continue to give benefit, specifically disease resistance. So I don't think it's that you shouldn't use them. I just think that thinking that you're going to change the population, specifically of fungal bacteria, I just don't understand how without changing the underlying soil conditions. Specific bacillus, I don't know. I suppose I don't really have a strong opinion there.
Well, if we're starting from a soil that doesn't have a lot of microbial diversity, it's not true soil. It's a it's a soilless media or potting soil or living soil, whatever you want to call it. Might have a compost or earthworm casting fraction. Would introducing microbes through a compost extract, a compost tea, these bugs in the jug or products like that have benefit? And I think the answer for me is sort of like a very confident maybe. Like, you might, it's worth testing in your environment, but I think, like you said, the environment plays the biggest role, more so than the product itself.
Sorry to interrupt, but if you're talking about a soilless media with a large proportion of compost, you're going to the microbial community is going to be coming in with that compost and be evolving, diversifying, sort of changing based on the the compost, really, and the amount of carbon that's labile, that's the sort of available to eat. So I would say if you're in a sterile like media, 100%, I think applied microbes would make a massive difference. But if you're using 20% compost or 30% compost and you're using multiple sources of compost, I don't know. I'm not totally sure. I guess I'm a maybe too.
Over time too, the plant is releasing exudates that's going to select for certain microorganisms over others based on, what you said, the various exudates it's putting out. So I think it's complicated. I think we know that. The other thing I wondered is, like, could we just water in a food source like fish hydrolysate, molasses, add a little alfalfa meal, whatever fertilizer that might be a good microbial food as a way of increasing this nutrient cycling process too?
So that's what I do. So every spring and every fall, I inject 2 gallons per acre of oceanic hydrolysate and humic acid and molasses. And to me, I'm feeding the primary purpose of that is as a soil conditioner to feed microorganisms, and to create a microbial flush, just a large increase in population. And it's not for nutrition, it's purely to feed them. So I'm essentially trying to feed the indigenous microbes in my soil. And again, that's sort of like taking advantage of what's already there.
Yeah, and I don't think I think this would be a really tough thing to research ultimately, because these microbe communities are so complex. I mean, we're looking at them from so far away, and they're evolving so rapidly that it's really, really difficult for us to kind of try and understand them or really make decisions around the whole process. So I think, like you said, we just have to look at it like as a whole, and then try to do what we can to take care of the populations, primarily through proper irrigation, starting with a soil mix that has good physical properties, both for growing plants and for these microbes. And again, temperature would be the other one, which is something that we can control in an indoor environment. Plays a huge role. The one thing I want to add now that I'm thinking about it, though, is when you do feed these microbes, you do have to be careful that they're not reproducing so rapidly that we're getting a thermophilic process happening that could actually heat up the soil, which your plant roots are not going to like, and potentially cause other issues, in terms of nitrogen robbing and things like that. Do you want to touch on that at all?
Well, yeah, I mean, that brings up an interesting point. When we're talking about living soils, I do think there's an argument where time is our best friend when it comes to microbial diversity, where the longer you have that soil, the longer the soil matures. And because what happens is, in that thermophilic reaction, is there's certain families of microbes that are spiking. And then when it cools down over time, that it's going to become a more complex profile, where other species are going to come in and start eating those families, et cetera. And I do think that the longer, and maybe I don't have a lot of evidence from of this other than just this is my philosophy. The longer you have a soil, the longer you use a soil and reuse a soil, the more diversity you're going to have. So that's what it made me think of. I mean, yeah, thermophilic reaction you don't want necessarily from a plant health standpoint, but I think as long as it stabilizes and you give it a lot of time, that'll sort of self-correct, and the microbes will sort of level out.
Okay. And I agree with all that. I want to go back a little bit, because there was a question from Norman asking about, in a synthetic setting, is it best to water in aminos and microbes on feeding days or plain watering days? And I'll let you answer that. But one thing I want to bring up with all this, it's important to know, is that there is a lot of research that shows that synthetic fertilizers, ionic salts, mineral salts in low levels feed microbes. It's a microbe food. They'll eat it. they'll cycle those nutrients. I mean, they don't need to cycle nutrients; they're already in ionic form, but it's not necessarily a negative thing from a microbial or soil health perspective in low doses. There's other reasons you may not want to use a bottle of nutrients or mineral salts for the effect on the environment and things like that. But from a microbial perspective, really low doses, and feel free to comment on this too. All the research I've seen shows it's actually beneficial.
Are those in? Do you know if that's in a soil system or like a soilless system, or what? Do you know what kind of media that would be in?
Yeah, that's a really good point too.
Because I think that would I would agree with that as long as there's available carbon. You know what I mean? Because microbes generally are limited by labile carbon and often by nutrients too. So any nutrient, even if it's salt based, will stimulate microorganisms as long as there's carbon there as well. Which in the soils we're working in, there's plenty because they're big carbon sponges. Just a thought.
Yeah, I'll have to go back and look. That's a very good point because the research doesn't always translate directly on things like that.
Yeah, it kind of depends. I mean, if it was like a rock wool media, I don't know if that would be the case, but anyway. Yeah, I think that's interesting. And I think that's an interesting point. I mean, yeah, one thing I think about all the time is microbes need a lot of the same nutrients that plants do. So their populations are often limited by something like sulfur, or in our systems, usually not sulfur, but could be manganese, could be zinc, etc.
Yeah. Now, if your EC gets too high, then you can deal with microbial loss through osmotic shock or osmotic pressure, and that's what's usually emphasized by the soil food web folks, is saying this is why mineral salts are bad for your soil health.
My argument is, and this is why when I make recommendations, I always try to point out that multiple applications tend to be better than one single large application,
yes, which
spikes your EC temporarily. So it's better to maintain consistency and balance of your soil solution EC. There's no doubt about it. And it's, in my understanding, is it's not that it just changes. It's very shocking on the population. So again, certain microbes are going to die, and then others will maybe thrive in a higher salinity environment. But if it goes back and forth, then you're getting lost on both ends.
Yeah. Well, I see a question here about cover crops, but I feel like that's a whole nother live we could do, just talking cover crops. We're getting close to that 30-minute mark. We have a few more minutes. Maybe if there's one more question you want to tackle, or did you have any other sort of like wrap up thoughts around microbes?
I mean, to me, just to touch on that cover crop thing, plant species diversity does matter. It's hard sometimes in our production systems, and I'm not necessarily saying that you should always do that, but I do think that is a really great general farming technique to maximize microbial diversity. But I think that what's more important that I've been thinking about more and more lately is how to maximize carbon flows through a system. Because again, usually microbial communities are limited by available carbon. So to think about large unavailable carbon, like, just think large particle size compost, for example, all the way down to amino acids and humic acid and fulvic acid, very, very soluble, immediately available. And there's a whole spectrum in between there. If you're constantly thinking about your carbon inputs flowing through a system, I think that is the key to maximizing microbial populations in whatever habitat you've given them.
I guess I've never really thought of it that way. Yeah, I always think of cover crops in terms of what is my reason? Like, what am I trying to accomplish with a cover crop? Am I trying to add organic matter because that's a limiting factor? Am I trying to sequester nitrogen? Am I trying to limit weed pressure? All of those things may be very different cover crops or combinations of cover crops, depending on where I live. And this is all for outdoor production I'm talking about right now.
Well, and I was thinking for indoor production, if you go through your goals, and I think, you've talked about this extensively, Tad, sometimes you end up with the decision that a cover crop doesn't make sense. Outside, I think that you want to choose the goal, but I mean, it depends on the production system, but usually you want to have something growing if possible. And I think like even, best practices now in South Dakota, corn, soy rotations are to just always have a living root in the ground. And so sometimes the purpose of a cover crop is just to have a root in the ground. And it's not necessarily a complicated multi-species mix. It's just a single species. And I think that really does support microbial activity because they love the rhizosphere. If you look at the concentration of microbes in the, 1 millimeter next to a root versus a foot from a root, it's orders of magnitude different.
Yeah, that's a really good point. I am all for diversity, but I think this idea that having two species of cover crops in the ground suddenly makes your soil more diverse than someone who doesn't have, has one species or maybe doesn't run a cover crop, at least indoors. To me, that's not really adding a lot of diversity. And I don't see a lot of evidence that that's going to make a huge difference. And this idea that we're trying to get closer to what nature does, it just seems like a very misguided principle to me, because nature would never select for this one crop to the exclusion of all others. Nature doesn't have weeds. Nature doesn't pull things out of the ecosystem like we do whenever we harvest a crop. So it feels a little like people are picking and choosing with this idea. It's kind of this like hippie mentality that, we're being more eco-friendly, but I just think that it's a little bit off base with what's actually going on when we're trying to grow a particular crop, especially indoors. When we talk about something literally called controlled environment agriculture, we're pretty far away from nature at that point. And there's practices I think that can be incorporated, but I want to look at each one individually and not just whether or not, and my bar is not what does nature do? It's what's going to be the best for soil health or plant health or optimize my yield or, whatever my goal is. So.
Yeah, I think indoors, that's a particularly good point. I think outdoors, however, when you're thinking about like how to design your agro ecosystem, I do think it's a little bit more relevant. And some of the concepts of what does nature do? I agree. We're not trying to replicate nature, but I do think there are certain things that are very simple and very basic that we should, take from nature. One being like, bare ground. If you can minimize bare ground, I think that's a good thing. And I think maybe it's just sort of a general framework for people when they're, thinking about certain concepts. But yeah, I agree with you. Inside, I think if you can do that to the detriment of your profitability very quickly.
Yeah, I think we're getting to the same point just from different angles. Because I agree with you. Outside, I think cover crops make a ton of sense for microbial health, for weed suppression, and all the other things that we talked about. So yeah, I'm certainly not against them. I just want, and I get in trouble for this on my Instagram, but I want people to think about the mechanisms behind why we do what we do, not just go through the motion of it, and start thinking about whether or not these assumptions that we've made are actually accurate, and why, the why behind it, I guess.
Yeah. I mean, to me, I think I'm real biased outside, because I think we should be trying to build organic matter all the time. And the best way to do that, the newest science shows that roots are essentially the best way to do that. You'd think biomass, like, oh, you grow a huge cover crop and you incorporate it into the ground, and that's how you build organic matter. It actually turns out that roots are how you build organic matter, and more specifically, microbial cycling and dead microbes and the interaction between microbes and roots. So to me, I do think people wildly overthink cover crops, and you're never going to replicate nature, and that you're never going to replicate a prairie. But if you have a root in the ground and you're capturing sunlight, like that is it. And the longer, the more time that you have something growing, whether it's a crop that you're going to harvest or whether it's a cover crop you're not going to harvest, the more organic matter you're going to produce, create in the long term. So that's why I'm like sort of biased. I'm a huge cover crop fan outside.
Nice. Yeah.
I
agree with you. I didn't realize the roots played such a role there in that process. So that's really interesting for me. Yeah. Well, I'll tell you what. So we're over 30 minutes. You and I just like to talk. So here's what we'll do. Everyone, if you're not following Bryant Soil Doctor on YouTube, please give him a follow. He's going to be posting other videos separate from the YouTube lives that we're going to be doing. So please follow and subscribe to him. And then if you have a question from this live, we're going to publish them on both ours. But if you want to get answered, write the comment on Bryant's YouTube. That's our way of getting people over there, because he just started his YouTube channel, and I want to try and support it. So if you comment over on his YouTube, one of us will answer the question. And I appreciate everyone that tuned in today.
Yeah, thanks everyone. Thanks, Tad.
Yeah, all right. I'm gonna start feeling better, and I have a propane guy right here because I have a propane leak, and I just saw my brother let him into my grow space. So I'm a little. I gotta go check out and see what's going on there.
Sure.
All right. Thank you so much, Brian. Talk soon.